Thank you very much. So the previous talks have given a nice introduction to some of the things I'm going to be talking about. So here is my lay introduction. As you've just heard, tumors are full of mutations, which are changes in DNA sequencing. We see patterns that emerge, which we call mutational signatures, when the mutations are analyzed across the entire sequence, which we call the whole genome. Some of these patterns can be attributed to environmental or exogenous agents, some to inherent cellular processes, endogenous processes. And it's important to point out that some of these assignments are actually based on informed knowledge rather than experimental detail. So what we set out to do some years ago was to say, can we generate mutational signatures experimentally, being an experimental group of scientists that might match those in tumors or in other ways give us insights into carcinogenic processes. So Ludmil's just told you there are 49 signatures, the 96 patterns of substitutional signatures, and that's relatively recent. When we set out to do this, he'd actually said there were 30, so he's expanded that number. So for the purposes of this talk, I'm going to talk about the 30 cosmic signatures from 2015, but the principles are the same. So here are the 30, and as I see it, what they, at the time we could assign some 9 of them to endogenous processes, summarized here. 7 of them to environmental agents, as shown here, and importantly, a good 16 of them unknown. So this is what we set out to do. Could we fill in some gaps? Could we show some insight into this? So this study was a collaboration between my group at King's and Serena Nick Zainal in Cambridge. And so what we did was we built up a list of as many of the known or suspected environmental carcinogens and some chemotherapeutic agents that we could think of, and thought, well, let's see if we can get mutational signatures from them. And so we came up with 79 of them were chemicals. We also had UV and ionizing radiation. And this is an overview of the study. What we did was we chose to treat, there we go, no, wrong pointer. Does this one work? No. You've got a pointer that works? Yes. Okay. Thank you. So we treated cells in culture. These were human-induced pluripotent stem cells. I'll come to that in a moment. Treated the cells, grew them up, sent them off to, isolated the DNA, sent them off to Cambridge for sequencing analysis. They undertook the computational analysis and generated, where we could see them, mutational signatures. But substitution signatures, dinucleotide signatures, insertion-deletion signatures. And we also looked at the general topography of where these mutations occurred across the genome. So there's about 6 years of work in this study, and I'm going to try and summarize it in 15 minutes or less. So I won't be able to cover all of this. So why did we look at stem cells? Well, they're generally stable, normal cells, normal in inverted commas, they're nearly normal. Importantly, they have indefinite proliferative potential, so we could keep growing them and keep growing them. And we could clone them. We needed to have clones in order to do the sequencing. So these were the key interesting features. There are some disadvantages. They don't generally have metabolic capability, so we have to add this in, because many carcinogens require metabolic activation in order to be active and to damage DNA. But we got around that by adding an exogenous metabolizing system. So this in outline is what we did. We found a dose or concentration that would that would kill around half the cells. We treated the cells for up to around 24 hours. We let them recover. We split them up. We derived subclones, and we sent these for whole genome sequencing. In some cases, we hit them with a higher dose just to see what would happen. And in general, I can tell you that we got the same signature at the higher dose as we got at the lower dose for those ones that we did it. So there's not a dose, it's not dependent on the concentration. And we looked to see whether we could detect DNA damage by a number of response elements here, just to make sure that we were seeing something. That wasn't wholly informative, but it was partially informative. So in summary, this is what we found. This is looking at the number of mutations that we saw in the clones for each of the 79 agents across here, plus a whole host of controls, solvent and so on, controls here. And every time you see the asterisks, we are seeing a significant increase in the mutational frequency above the background. There is a background one, I'll just come to that in a moment, and in many cases, in about half the cases, just over half the cases, we got a signature, and in the other half, we didn't. So it wasn't an entire success, but it's a case of glass half full or glass half empty, however you want to look at it. We got quite a few of them to come up, so we were very pleased about this. So we did see this background signature, the so-called cell culture signature, and it looks like this with the 96. And it turns out this is really quite similar to one of the cosmic signatures, signature 18. And I'll show this again a little bit later very quickly, but this has been assigned speculatively to reactive oxygen species. And that would make sense because we're doing tissue culture at 21% oxygen, and that's quite high, and that does stress cells. In vivo, in the body, the cells in your body do not experience oxygen levels that high, but tissue culture is generally done at that high. So we saw this every time we isolated DNA from a culture, treated or untreated. We could see this signature, either the only signature or a background signature. So we have subtracted that from all the signatures, and in 41 cases, we still saw a distinct mutational signature. I'm going to quickly go through some of these. So there were 3 cases in which we got a very good match with the human tumor signatures. And the first one of this is aristolochic acid, which gives this predominantly T to A mutations, virtually nothing else, the most common one being in the CAG context. And this, of course, matches the cosmic signature 22, which has been assigned to aristolochic acid. So a very good match there, very, very pleased to see that, good confirmation that we have a system that works. And the second one, again, quite familiar one, not unexpected, was comparing simulated solar radiation, which contains some UV, which gives us a signature very similar to the cosmic signature 7. And as Ludmil explained, there's now several subsignatures of signature 7, but in this case we get a good match. And all these mutations, most of the main mutations are occurring at pyrimidine dinucleotides, which again is what you would expect from the chemistry of UV damage to DNA. So a very good match there. And then the third case is looking at the polycyclic aromatic hydrocarbon, benzpyrene, and also its reactive intermediate, benzpyrene dialipoxide, giving a signature that looks like this, predominantly showing C to A mutations, and this looks very like signature 4. Not quite exactly the same. You'll note there's a big peak here in the T to A region, which you don't see with benzpyrene. We wouldn't expect to see it with benzpyrene because we know chemically that there's very little reaction with adenine in DNA, but we have some suggestions I'll show you in a minute as to where this one might come from. So a good match, but not a perfect match. We kind of wouldn't expect that all the smoking signature would be manifested in one single chemical. So, and I'll show a bit more about that in a minute. But a good match anyway, exactly what we had found before with other systems. So if we do a cluster analysis on the 41 mutational signatures, that's shown here, and it breaks down into about 9 clusters. And this is looking at where the polycyclic aromatic hydrocarbons, and we had quite a number of them, where they cluster. So you've got a whole group here, all clustering close together, these ones here, and then you've got one up here that's dibenzo-AL-pyrene, which is quite different from the other group here. And that, again, we can understand why that is the case. And here I'm showing 3 of them, benzpyrene and its dialipoxide, dibenz-AH anthracene and its dialipoxide. These 2 are very similar to each other. And this one, dibenz-AL-pyrene, which is quite different. And that's explained because this compound reacts predominantly with adenine residues in DNA, whereas these 2 much prefer to react with guanine. And you can see that reflected here. Here are the mutations at C to A, or G to T as it is, and here they're at T to A or A to T. So this peak here, which I just showed you, is also seen in tobacco smoking, signature 4. So it's coming up here. It's another PAH, not seen with benzpyrene. But a note of caution here is that if you take simply this portion of the signature, the T to A signature, this looks very, very like the aristolochic acid signature. So it's not necessarily the case that these things are giving different signatures. We're seeing some overlap here. No one's suggesting that dibenzo-L-pyrene is responsible for aristolochic acid nephropathy and associated neoplasia, but it's a caution that you can see signatures that are similar from rather different agents. So another case of where we see similar things, two platinum drugs, carboplatin and cisplatin, they cluster very closely together, and here are the signatures, both of them showing predominantly a GGG signature and an AGG signature. That's what we might expect, because although these chemicals are different, the way they react with DNA is exactly the same. So you would expect the same signature from both of them, because the damage that they cause is the same, and that's exactly what you find. Cosine similarity very high, 0.95. And then looking at alkylating agents, here we see two distinct clusters, and it doesn't matter whether it's an ethylating agent or a methylating agent. So here we have dimethyl sulfate and diethyl sulfate giving us signatures up here, and then this group here, temozolomide, which you've heard about, and methyl nitrosourea, ethyl nitrosourea, and dimethyl hydrazine, all clustering close together. And here there are a few surprises. So here you can see the top two that I mentioned, giving mostly signatures here in the red, but with ENU, MNU, and temozolomide, the signature is dominated by T to A here, which is not what we expected, particularly if you take the so-called temozolomide signature from the COSMIC, which is giving predominantly C to T. So we're seeing a shift. We're not seeing a good reproduction here. Even though temozolomide, MNU, and ENU are very similar, they're not giving us the expected signature 11. We're not quite sure why this is, but we've got a few ideas. There may be DNA repair processes that are overactive or more active in the stem cells that are giving us a shift in mutations, but that will require further investigation. And then, so the signature 18, which I told you was thought to be due to reactive oxygen species, here's experimental proof that it is, because when we treated the cells with potassium bromate, which generates hydroxyl radicals, we got a signature that was very similar to signature 18. So here's, if you like, first experimental evidence that signature 18 is in fact due to reactive oxygen species. And then we also look for dinucleotide substitutions. Ludmil described these briefly. These are much less frequent than the substitution mutations, but a number of the agents gave it, specifically and interestingly, as we might expect, the solar sunlight giving quite a high level of CC to TT, and then low levels for some of the PAHs here, giving the CC to AA, and then with the carboplatin, with the platinum compounds, we're seeing two types of disubstitution. So a much lower level of these, but nevertheless distinctive patterns. And then we also saw a small number of the agents also gave levels of insertions or deletions, and these are shown just briefly here. I won't go into them in detail, but we have again a number of the agents, cisplatin giving this signature, and some of the polycyclic aromatic hydrocarbons, and some of the nitro polycyclic aromatic hydrocarbons giving signatures like this. So this is a summary, if you like, of the 41 that gave substitution mutations, some with lesser confidence, shown in light blue, and you can see that a few of these agents also gave either disubstitution signatures or indel substitutions, or in some cases both. So quite a mixture here. And it's interesting from a technical, mechanistic point of view that we can see different types of signatures from the same agent, and this is giving us insight into how DNA is damaged by these agents and what systems, cellular systems, may be acting on them to attempt to repair them or fail to repair them and end up with mutational events. So overall, it's a mixture of the expected and the unexpected. Cisplatin and carboplatin, we expect them similar. These three are similar, but not what we expected. Again, pH is similar. We also saw very dissimilar signatures from compounds that are very similar. Here are two nitro-PAHs, and here are two forms of aristolochic acid, giving dissimilar signatures. But we also saw dissimilar agents giving similar signatures, so we have to be careful how we interpret these things. Coming towards the end. So this is all very interesting in itself, and from an experimental point of view, we've got a lot of new data to understand mutational events, but how do they interact with the human tumors? And so what you're seeing here is the similarity with the cosmic signatures here against the agents that we looked at. And the darker the red, the closer the correlation. So you can see there are quite a number of agents with some similarity to signature 4. Many of these are present in tobacco smoke, but some of them are not. So again, we have to look at this cautiously. There's aristolochic acid with signature 22 coming up nice and red, but also this PAH is giving us an anomalous color there. And temozolomide is not giving us a match with signature 11 over here, but it's giving us some matches with some of the other signatures, and we have to understand what that's about. And then if you look tissue by tissue, then you see a large number of matches here with head and neck cancers to do with smoking, and a few here with the UV up here, and then quite a number matching up with the endogenous reactive oxygen species process over here. So very quickly, that's a summary. Here's my lay summary. We have generated whole genome mutational signatures for 41 agents. This comes to 853 signatures, base signatures, taking into account we have different concentrations in some cases, 8 dinucleotide signatures, 10 indel signatures. Some are identical to human tumor signatures, and many others are similar. So this is a good start. This is a reference set of signatures, which we think is a new resource to understand mutations and to search more into the future for links between environment and human cancers. And just finally, to acknowledge the people who worked on this. Here they all are, some from my lab and the others from Cambridge, and we published this a few weeks ago in Cell. Thank you very much.